So first of all, I would like to thank Professor Luc Montagnier for the kind invitation. It's really a pleasure and honor for me being here in such a wonderful audience. I would like to focus on a field that may look a little bit provocative, but I hope it will not look like this at the end of my talk. All the universe and living systems are permeated by mutant vibrations and rhythms. And it's now increasingly becoming evident that even in living systems, the rhythmic synchronization of oscillatory patterns underpins wide-ranging programs of gene and protein expression, ultimately driving cell growth, differentiation, and even embryo development. The issue of sound rhythms brings us to the idea of music, but not how we perceive music in modern times. It's not simply the skill of the artist to play an instrument or to emit a wonderful voice. In ancient times, musica techne meant basically the effort of the human being to perceive themselves as unique parts in the universe and to look for a rhythm as a measure, a number, and a weight for such a relationship. And if you can see it, if we look at the cells and you look at what we call the cytoskeleton inside the cell, it looks like a fairly static environment. But if we use other techniques, fortunately there's lots of light, so probably it scatters, you cannot see it as I would like to. But if you look at these images, when we were able to record the motion of the cytoskeleton inside the cell, this is a human fibroblast, you can see that everything is pulsating and vibrating in the cell. And if you combine confocal microscopy and atomic force microscopy, and you give a look at what the cytoskeleton looks like, then you see a continuous, constant motion, vibration. So you can actually see that elements from the cytoskeleton are moving from the cytosol to the nucleus, and vice versa. You can see that each element, this microtubuli, is intrinsically vibrating. And you can see that there is an intensifying and rarefying of the network that you find inside the nucleus. It's kind of scaffold is vibrating. So the DNA is also vibrating. And our current view as a molecular biologist is changing. What we call molecular signals, protein kinases, transcription factors that change the pattern of gene expression in cells are not simply swimming inside the cells, but they somehow walk on this network, this physical vibrating network by the aid of molecular motors like kinesin or dyneins. And so it's important that the molecular recognition patterning is not simply, and it cannot simply be a lock and key model, but it's also phase resonant mode of interaction, this physical interaction that occurs on this vibrating network. You see, even a plasma membrane on your right is not stable. When we talk about growth factor interaction with receptors, we have to figure out this kind of inherent vibration that we can actually measure by the aid of techniques like atomic force microscopy, for instance. These are other examples. Even intracellular mediators like intracellular calcium, you can see how it moves from the cytosol to the nucleus in neuroblastoma cells, for instance, due to the presence of a growth factor, an air growth factor. But once again, at the closer look, when by confocal microscopy analysis you follow the three-dimensional pattern of calcium oscillation in the cytosol, you see on the right, it's a pattern of vibration. Here, investigators have been using a synthetic calmodulin, which is a calcium binding protein, which oscillates, you can see, in phase with the oscillation of intracellular calcium during the cell cycle. And what about fertilization of a oocyte, which is the first step to generate a totipotent stem cell? Once again, the calcium is oscillating with specific particular dynamics. And if we use calcium chelators to block this, and then we transfer these fertilized oocytes in mice, we won't have any embryo anymore. So this is important to keep in mind that somehow we have to stop looking at the cell biology only on the perspective of chemistry, but we also need a closer look at physics. Since long time, as you can see here, 2001, indeed are we able to follow quantum spots related to intracellular, for instance, calcium signaling, and to see what each kind of spike, what we call spike, is made by the merging of smaller entities like sparks, and even smaller entities like sparklets, and something which is apparently noise at the end of the story, but it's a kind of scale-free field for information. So when we move to stem cells, that are very nice because basically they do not look like any cell of the adult body at the beginning, but they can become any kind of cell. They can form a complete individual. We used to look about this very old scheme, starting from totipotent cell, which is so far is only considered as to be the fertilized oocyte, because a singularity can give rise to an entire individual. Then we have downstream what we call pluripotent stem cells, so-called embryonic stem cells. They can give rise to any cell of the adult body. They can contribute to the formation of the entire individual, but there is no way that a single cell like this can generate an entire individual. That's why we call them pluripotent. And then multipotent cells, so-called adult stem cells, that are residing basically in any tissue of the adult life, even in the heart, in the brain, in any tissue. We call tissue-resident adult stem cells. So the dogma was that you should not go in the reverse mode. From a multipotent cell, you cannot create a pluripotent cells or an embryonic-like stem cells. Or, even worse, from a somatic non-stem adult cell, like a skin fibroblast, you cannot generate an embryonic stem cell. But this has been definitely challenged, and the Shinya Yamanaka, John B. Gordon, they got the Nobel Prize in medicine or physiology because they found that simply by delivering a few set of genes inside a somatic non-stem adult cell, even a human somatic cell, you can revert this cell backward to a pluripotent embryonic-like state. That means creating our own autologous embryonic-like stem cells. Now what I'm going to show you that we can do the same story by using physical energy and properly delivered magnetic fields or vibrations without the needs of viral vector-mediated gene transfer. That means the clinical application is closer, especially because I'm going to show you that we have been using these strategies on kind of human adult adipose-derived stem cells that we are already using in clinical practice as doctors for treatment, for instance, of diabetic foot and osteo or cartilage disorders. So one of the first evidence that you can orchestrate stem cell patterning, not only by chemistry but by physics, came from this work that we have performed in mouse embryonic stem cells by using extremely low frequency magnetic fields. Magnetic fields, they were 50 hertz and 0.8 millitesla. Basically, the same kind of magnetic field that you find in the power line, in the domestic power line. In those years, we also discovered the master gene of cardiogenesis in embryonic stem cell, which encodes for a small peptide, which was originally thought to exist only in the brain, was an opioid peptide named dynorphin B. And the preprodynorphin gene was found to be the master regulator in embryonic stem cells to drive efficient cardiogenesis. And I hope you can see it. Can you see? On your left, the picture which is contracting. There's too much light scattering on it, but I hope you can see. Otherwise, you're welcome to come on my computer and see it. And basically, if you look at the cardiogenesis, it's very challenging. Not only because it's one of the most spread and terrible diseases for human beings. Most of humans are dying because of cardiovascular diseases, but it's also challenging because cardiogenesis is only 0.002% of the overall differentiating potential of the inner cell mass in an embryo, even in the human embryo. That means that if we want to succeed with a good cell therapy for heart failure, we need to enforce the process of cardiogenesis by different orders of magnitude. So on your left side, you see that we basically transformed a carpet of mouse embryonic stem cells into a micro heart. It's a spontaneously beating micro heart in vitro. But you can see on the right side, there's no way to avoid such a strong light on. Just for a moment. No? Okay. Can you see it's contracting on your right? I'm so sorry, but if you can turn the light off for a moment, please. Oh, there is light coming from there. No way? Sorry. Okay, if you want to come close to the computer, I can turn it and show you. But this is very important. The same effect can be gained by exposing stem cell to electromagnetic fields. So then we reasoned that we may probably set up a new system to expose a target biological system to a magnetic field. And what we did is the development of this novel apparatus called radioelectric asymmetric conveyor. This is emitting at Wi-Fi energy, basically 2.4 gigahertz. The radiated power that you find on the card on your computer when you connect to internet is about 12 milliwatt. The machine is emitting at 2 milliwatt. As warm bodies, as living organisms, human bodies emit around 58 milliwatts radiated power. And what's important, you see, this cable here, we call it asymmetric probe because it's not sending any magnetic field to the biological target. It's simply sampling, holding, and giving back to the tissue its own radioelectric field that is modulated by the incoming electromagnetic field produced by the machine. So it's well known that when you expose a biological target to an electromagnetic field, you are inducing microcurrents. So these microcurrents are going to be taken up by the probe, sample, hold, and given back to the target. So the cycle is 250 milliseconds and 5 seconds interval. So what we've done is, by this system, we exposed it only for 72 hours mouse embryonic stem cells to these, we call asymmetrically conveyed radioelectric fields. And we optimize it by several orders of magnitudes, the ability of these cells to get committed to complex differentiating outcomes like cardiac development, like neurogenesis, skeletal myogenesis, and vascular commitment. And I'm going to be blind of this one. And I'm really sorry, but I think it's impossible to see anything with such a strong light on the screen. So what? There's no way to turn it off. Okay, but in these screens over here, if you some of you will come and see it, so you can see the difference on the screen. Okay, so tubulin stays for neural commitment, myod and myogenin stay for skeletal muscle differentiation. And the other two markers on your right, MAC and alpha-actinin, highlight the cardiogenic commitment. What's important, it's not only the increase in cardiogenesis, but even the quality of the commitment. If you look at the bottom panel on your left, when it says control, then you see that the contraction, spontaneous contraction, is a kind of scattering. If you look at your right side, when it says REAC, the cells that have been treated with the magnetic field, they do exhibit a pacemaker-like activity that tries a wave loop form of contraction. This is very important, but it resembles very much what is really going on in a living heart. Then we were able to do basically the same thing with human adult stem cells that were harvested for the adipose tissue. The same kind of cells we were authorized to put into clinical practice, especially for peripheral vascular diseases. And this is again the paper that is going to appear soon in Cell Transplantation. And what is important by exposing again for a brief period of time, only 48 hours, these cells to the asymmetrically conveyed radioelectric field, we were able to trigger the expression of genes related to cardiogenesis, vasculogenesis, neurogenesis, skeletal myogenesis, and most important, we were able to transiently activate genes that we call pluripotency genes. These genes are important because they set the cell backward to an ancestral state where we can say, okay, the cell is pluripotent. The cell is able to do things that in the adult period of life, even if it is a stem cell, is not able to do. So this is why, basically, once again, it's better if you look at the screen over there. The green part highlights the finding that by exposing these cells to asymmetrically conveyed radioelectric field, you have a strong commitment to cardiac development, alpha-actinin. MyoD in the center is for skeletal myogenesis, and on your left side, tubulin is for neural differentiation. As you see in the upper part, the unstimulated cells, they only exhibit a blue spot, which is the nucleus, without any expression of tissue-specific marker for the differentiation. And now, as you know, these two outstanding investigators, they got a Nobel Prize for reprogramming human adult somatic cells backward to a pluripotent embryonic-like state, and but they had to use viral vector-mediated gene transfer in order to induce such a reprogramming. That means that this is not really readily transferable into clinical practice for many reasons. Also, because at the end of the story, they freeze the cell into an embryonic-like intermediate, which has only an extremely low ability to transdifferentiate into other cell types. So basically, you're going to have 95 to 98% of embryonic-like stem cells and only 0.0 something% of cells that are committed to cardiac, neural, or other important lineages. And so the tumorigenic risk is very high. But what we did is using the REACT technology, once again, we delivered asymmetrically radioelectric fields to human non-stem adult somatic cells like dermal skin fibroblasts. And what we got was a transient upregulation of pluripotency genes. It is important that it's transient because these genes need to be upregulated in order to make the stem cell or the adult somatic cell pluripotent. But then they need to be shut down in order to allow a proper differentiation. If you use a viral vector-mediated gene transfer, you're freezing the cell into a kind of pluripotent intermediate, and there's no way to get this cell going through a high throughput of commitment to any kind of differentiating outcome. And you can see the difference. About 10, 15, 20% of each lineage can be achieved in somatic adult non-stem cells, human dermal skin fibroblasts, by exposing these cells to radioelectric field. Basically, you're going to be able to commit these cells to a destiny that they will never otherwise acquire spontaneously. We were also, this was just published in scientific reports, the front journal of Nature, just a few weeks ago. We were also able to reverse the stem cell senescence, so the molecular senescence that occurs in stem cells when they undergo prolonged culture in vitro. And this is needed in regenerative medicine in order to have lots of cells to be transplanted. So basically, we used to keep these cells in culture for several weeks, even 10, 12 weeks, in order to have 400 million of cells that can be transplanted in any given protocol. But in this way, we are inducing aging in these cells. So we are losing part of the cell ability, the stem cell ability to rescue a damaged target tissue. By this way, we can revert these cells backward to a young phenotype and once again somehow reawaken their ability to be committed to many different types of differentiating outcomes. Not only physics in terms of electromagnetic field, but even sound vibrations. Several years ago, in collaboration with James Ginseski, physicist at the University of California, Los Angeles, who was a former co-worker of Richard Smalley, who all know, developed a technique was based on the use of atomic force microscopy, shown here. Basically, it's a cantilever with a probe, carbon nanotube ending with a single atom. So you're touching the cell and you're creating a atom contact mode, one atom from the probe and one atom from the counterpart, one atom from the cell. So if you're moving in a pressure constant contact mode on a cell, even a single molecule is going to look like a mountain. So it deflects the cantilever, and by projecting a laser beam on it and relying on the deflection angles of the laser beam, then you can create a map of actually seeing, well, I will say better perceived instead of seen, because even the strongest conventional microscope by its own lenses is a projection of human eyes. This is going to be a projection on the finger of a blind man. So it can also measure inner and physical forces on the system. And I hope it works. By this system, we were able to record vibrations from yeast cells, noticing that the pattern of vibration was changing according to the kind of substrate that was present in the medium, or according to the fact that by keeping the substrate constant and changing the temperature, we were varying the transcription rate of given gene program, gene patterning. And this resulting in a dramatic change in the type of nanomechanical vibration that can be recorded at the cell surface. So Can you hear me now? So the idea we came up with was to proceed in doing a sort of interrogation on nanomechanical signatures of stem cells undergoing a given commitment. That is, we can record different vibration patterning, different sound patterning from stem cells undergoing, let's say, cardiogenesis, neurogenesis, or any other commitment. And the sound vibration is totally different according to the lineage commitment that these cells are going through. And then expose undifferentiated cells to this kind of nanomechanical patterning to achieve a specific commitment toward a given lineage. You can imagine that this is totally revolutionary because you're not using synthetic chemistry anymore or viral vector-mediated gene transfer, but only physics. And we also patented these strategies. I'm concluding very soon. There is now evidence that proteins are vibrating, but not for just a few microseconds. When this story was looked by different technologies like terahertz near-field microscopy, it turns out that a chemical bond can vibrate for many hours, which is enough to establish phase relationship among different partners. And a wonderful example of a sound emitted from the organ, from an organ, is the heartbeat. And we always think about the heartbeat like tutum 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 tutum, something like this. But we can go further. We can go further by phonocardiography. We can dissect the sound and the pitch coming from the heartbeat, and we can gain information about a normal heart with some, we say, wavelet transform of the phonocardiogram, or we can see how the pattern of vibration changes when we have a post-infarct heart failure. But the nice story I'm going to tell you, and I will conclude with this, is a story of a dear friend of mine. He's a famous jazz musician, Milford Graves. He's a legend for people who love jazz and drums. And he finally gets sick with arrhythmia, and he said, Jesus, I spent all my life on rhythm, and I don't want to die because of rhythm. So he started to record his own heartbeat with an electronic stethoscope and make a comparison with other people with a normal heart. And what he did was something that a scientist will never have done. Just looking at the subsonic part of the vibration of S1, the first tone of the heart, was around 5 hertz. So he rose the pitch many times up to uncover that tiny part of the sound of the heartbeat entailed a micro rhythm. And so we have this kind of rhythm inside. When we say heartbeat, it's not simply tutum tutum. It's a kind of rhythm, and by administering this vibration, or mechanically, or by electronically, by a needle connected to an electronic trace of the sound that you heard, which is electronically reviewed as by a lab view patterning, he was able to heal from his own arrhythmia. He was not believed at the beginning, but at the end of the story, other physicians and scientists with the same problem used this system, and they improved it dramatically. So when I met him, I said, why don't we try the sound of the heart on the stem cells? Especially because I was intrigued by bus at the maximum. Okay. These are the individual 8 frequencies that he recorded. This is the first one. Imagine that we have this kind of mechanical transducer to expose stem cells to this sound, this pattern, these microrhythms. And we were able to finally tune the expression of pluripotency genes, SOX2, OCT4, NANOG, the same genes that Shinya Yamanaka elicited with viral vectors. The expression was only transiently induced, so we came up with a direct reprogramming of the cell. The cell, these are human adult mesenchymal stem cells. They become transiently embryonic-like stem cells just for the time needed to acquire a pluripotency state. Then spontaneously, these genes are downregulated, even during the exposure to the sound of the heart. And at the end of the story, they do nicely express genes for the cardiac commitment or for the vascular differentiation. And this is what the final picture looks like. Once again, it's better if you look at the screen over there. You can see nice commitment to myocardial cells and vascular cells. So concluding, I'm really moved when I talk about Emilio. And together, we did some experiments that are in this line with Emilio Del Giudice, Alberto Tedeschi, and Giuseppe Vitiello. I say they are all here, even Emilio. And what they have done is just putting leaves, so vegetal cells in water, stressing these cells and transmitting some signature, some signaling information to water that indeed acquire a more ordered state, as shown by their calorimetric measurements. And they also transferred this kind of more ordered state to a solid substrate like plastic or glass that was initially warm and then cooled inside this liquid. Then the plastic or the glass was broken in many parts. The solution was stirred by the mechanical collision among these parts. They recorded a sound from which they skipped the non-fractal part, only maintaining the fractal part of the sound. And so they created an acoustic trace. They took the volume off only by leaving the electromagnetic field that's moving the solenoid that then makes the loudspeaker membrane to vibrate. And once again, this is nice and important. These are preliminary experiments, but we have an NO4 performing in triplicates. We can once again move pluripotency gene and cardiac and vascular gene. So it seems that whatever you do to talk with stem cells and somehow convince these cells to acquire a coherent state, they do respond to going backward to an ancestral pluripotent state. They can remember what they just forgot throughout the adult life. And I would like to conclude with what we wrote several years ago with Jim Ginczewski. As we say that both in philosophical and the visual sense, seeing the equation, seeing is believing, does not apply anymore to nanotechnology. Today I would like to say to this extreme field of molecular biology and physics, for which there is nothing even remotely visible to create a proof of existence. When we move on to an atomic and molecular scale, data are recorded by sensing and probing in a very abstract manner, which requires complex and approximate interpretation. More than in any other science, we need a novel paradigm, which is a narrative and information, which is the essence of life. So I thank you for your attention. I would like to thank all my co-workers, and I hope to have touched your heart.